Chapter 2, Basic Principles, Parasite Systematics, Ecology and Evolution, Section Parasite Ecology, Part 1. When we look at the ecology of a parasite, when I say ecology, most students think out there in the wild, the streams, the lake, the woods, the ocean, whatever. But you also have to keep in mind, we are the primary ecological niche for many of these parasites we're going to be talking about. The ones that are obligate, endoparasites. The ones that are going to be, facultative, ectoparasites. A tick doesn't want to be sitting on a leaf. No, a tick, a flea, wants to be on a host because there it has food, it has warmth, it has some protection from its own predators. So we have to keep in mind, we are part of the parasite's ecology. So keep in mind, like I said, that somewhere on us, somewhere in us, is the ecological niche for that parasite. Not saying that the parasite could be anywhere in us. No, they have evolved to maximize the availability of nutrients, water, and all of that, and survive within a specific place in our bodies. Okay, you can't take a parasite and just put it in anywhere within our bodies, within a domesticated animal or a wild animal, and expect this organism to survive. Great examples, as you see here. Ch2 Americanus, hookworm. Okay, hookworm will die if it is ingested. Okay, boom, within seconds it gets digested away as part of your normal meal thanks to the stomach acids. But put it subcutaneously, and Ch2 Americanus is going to survive, is going to do extremely well. Flip side, Taenia saginata, what we think of as a tapeworm. Part of its larval development is the passage through the stomach. That is going to trigger further larval development. So what Ch2 would be destroyed by is part of the signals required for further development by Taenia. But then again, if Taenia somehow got into a wound, the larvae got into a wound and it got under the skin, it would be dead in hours as our immune system destroys it. Every parasite has a host, and within that host, it has a niche somewhere there. This is cutaneous larva migrans, hookworm, and a few of the others that like to get under the skin and go on walkabout. What you see there, those little lines that are on this patient's foot, those are worm tracks. Ch2 can do that. Taenia, not so much. What's driving this niche within the human host, within the domesticated animal or the wild animal host, is not only nutrient requirements, but other requirements that need to be met for survival of that parasitic species. Things that are going to trigger further larva development. Could be, like I just said, with Taenia, passage through the acidic environment of the stomach. Turns out the stomach, more highly acidic than the small intestine, and stomach definitely way higher than what you're going to find in the large intestine. For some of the, like the liver flukes, it's going to be the availability of bile, the bile salts specifically. It's going to trigger, locomotion of the larvae of the developing fluke. It is going to go up the concentration gradient into the bile duct, into the gallbladder, into the liver. What we're also finding is that the intestinal parasites, the tapeworms and the nematodes and things, are just because they want to be in the gastrointestinal tract doesn't mean it's willy-nilly. Turns out the gastrointestinal tract, small intestine, large intestine, has regions within those intestines that have slightly different microenvironments. The ascending colon, transverse colon, descending colon, each have their own jobs. Slight modifications in the environments, which is done, all of that's going to be completely different than what you see in the small intestine, at the duodenal, into all the different areas. And that's just with us humans. You have to take into account also the other animals out there, the other mammals, the lizards, although I can't think of any animals right now. But every animal is going to have a slightly different environment in the different regions. And it turns out that the intestinal parasites are going to migrate, and they're going to, as they're being pushed through the intestinal tract, thanks to, peristalsis, they're going to be moved along until they hit the environment that is perfect for them, and that's where they're going to stay. How do they stay there? Well, as we'll see later in this semester, all the different parasites have their own means for staying where they want to be. And you look here at this figure here from the textbook. They're showing different cestode parasites that are found in birds, specifically aquatic birds. And they're showing that many of these parasites have much broader ranges of where they will want to be. You look at the top one there, what is that? Tetraborithius. Tetraborithius is going to be found 80 to 90% of the gastrointestinal tract. So that one has a wide range for its ecological niche. Where you go and you look at the bottom one there, what is that? Diarchus? These are bird ones I'm not familiar with. Diarchus. Its specificity is such that it's less than 10%. It's going to be found in less than 10% of the gastrointestinal tract, which means it has a very narrow range that it wants to be in. Too hot, too cold, just right. Too acidic, too basic, just right. Too high salts, too low salts, just right on the bile salts. And we have to keep in mind that the environments within the gastrointestinal tracts between species, between genuses, between vertebrates is going to be completely different. You look at this picture here, this artist rendition. Now, keep in mind this is a generalized rendition. It doesn't show the complete lengths, but it gives you an idea of the relative lengths. Our human gastrointestinal tract is relatively shorter compared to that of a rabbit, because the rabbits are eating plants that are going to be high in hard to digest cellulose. They have to eat a lot of it, and they're going to need more time for the digestion of it. Whereas we humans, yes, we eat some fibrous material, but we also eat a lot of meat, and we also eat a lot of dairy and grains that are quick and easily digestible. So we really don't want to keep it around that long. So we want it to get in, get digested to a certain point, and then we're going to pass the rest out in our feces. Whereas rabbits, what they're eating is very low nutrient dense. You know, they're not always going to be able to get the carrots and things like that that we feed them. A lot of times they're just eating grasses. And there's a reason why ruminant animals, as you look there down at the bottom left, they're showing a deer there. There's a reason why ruminant animals like deer and cows and sheep and all those have to eat all the time. Turns out the grasses they're eating are low density nutrient value. You look, you know, the bottom right there, that fox. Fox is going to be eating primarily carnivore. Turns out foxes are like us. They're omnivores. They'll eat some vegetation, you know, if that's all there, but they're going to choose, given a choice, they're going to want to eat that vole up at the top left because the meat is nutrient dense. So just like us, their gastrointestinal tract doesn't have to be as long. What's the point of these past two slides? It's to show that the gastrointestinal tracts are not the same for all mammals. It's not going to be the same for all animals. And even if they, you know, like the deer and the rabbit, you know, they're relatively close to being in length-ish, there's going to be lots of differences that are going to increase the chances for more ecological niches within the GI tract. More niches, more chances for different parasites to flourish in these different regions.